Buenos días, bienvenidos todos al workshop de BIKO. Estamos acá con el doctor Keating, que nos va a presentar un nuevo producto de Hibergene. El nuevo producto de Hibergene, que es un producto más rápido para la determinación de virus y bacterias por una tecnología nueva, que es la tecnología LAMP. El doctor Keating es un PhD en el campo de los inmunoensayos y los biosensores, y es de la Universidad de Dublín. Tiene 20 años en manejo de empresas que tienen investigación y desarrollo, como Abbott, Bioquímica, y ha trabajado como director de tecnología en la empresa Hibergene desde hace 6 años. Ha generado nuevos productos que tienen marca de la Comunidad Europea y que sirven para la detección de enfermedades infecciosas. En el día de hoy nos va a presentar una forma de detección de esas enfermedades infecciosas y virales por una tecnología nueva que es la tecnología LAMP, y él nos va a hablar de las virtudes de esa nueva tecnología. Welcome, Dr. Keating. Thank you very much to be here today. Thank you.
Thank you, Patricia, and good morning. Thanks for the opportunity to speak to you today about Hibergene's range of fast and accurate LAMP-based molecular detection products. Just a little bit of background, first of all, about Hibergene. We're a small Irish company based in Dublin, established since 2014 here, and we develop on-site, manufacture and market from our Dublin base a range of 13 infectious disease molecular diagnostic tests, all of which are based around LAMP technology. Our site is ISO 13485 certified. All of our products are CE marked and adhere to the CE marking requirements, but we sell products globally through a range of distributors. And in terms of our South American presence, we're currently registered in a range of countries you can see there, Brazil, Colombia, Venezuela, Peru, Chile, and in the process of registering for a number of others. So we're a small company to a certain extent with a global footprint. A little bit about our product range before I go to the detail of how these products work and how we harness the speed of the LAMP technology across this range of diseases. So they split really across 4 different areas at the moment. Clostridium difficile or Clostridioides difficile is our only presence in the healthcare associated infection. We started off developing and manufacturing tests for bacterial meningitis. So we have a large range of products which detect Neisseria meningitidis from a range of different sample types. And we also have a combo test which detects both pneumococcus and meningococcal bacterial meningitis. And then also a test for group B streptococcus. We have a range of sexually transmitted infection tests, which include chlamydia, gonorrhea, herpes, and mycoplasma genitalium. And also a large and growing portfolio of tests for respiratory infections, mycoplasma pneumoniae, flu, RSV, and obviously, and most recently, COVID-19. And I'll come to that at the end of the presentation in terms of our COVID-19 test performance. But I really want to come back a little bit and talk about LAMP. Obviously, it's at this stage a reasonably mature technology in terms of the amount of time that it's been published and it's been used. It's obviously also something that's become quite prominent due to the COVID testing landscape and the requirement in many applications for rapid molecular testing of COVID samples. So just some very basics on LAMP. It's a sort of an acronym for loop-mediated isothermal amplification. You can break that down into the 3 parts. The loop-mediated refers to those loop structures that are formed, which are the basis that allows the isothermal amplification to take place. The tests are isothermal, so typically require simple instrumentation taking place at a single temperature. In our case, for the Hibergene assays, that's usually about 63 degrees. And the level of amplification associated with the loop-mediated isothermal amplification is quite rapid. We typically generate very large quantities of amplicon in quite short periods of time compared to other methods. So the name itself really expresses a number of the key elements of the technology. If I talk in a little bit more detail about the advantages of LAMP technology, and the structure on the slide here is just the basic starting dumbbell structure. So essentially, LAMP technology requires the identification of ideally 8 conserved target regions on the genome, and then the production of 6 primer sets which cover those 8 different regions. And by designing that set of primers correctly, you end up with this dumbbell structure at the end of the first phase of amplification, which becomes a very efficient self-priming template for the reaction. So in terms of performing the test, and in terms of developing and marketing the test, there are a number of obvious advantages to LAMP. There's no thermal cycling required. That allows LAMP tests to be performed on simple equipment. That can be as simple as a water bath. In some publications, our test uses a very tightly controlled thermal block with an optical system. Those multiple primer binding sites mean that there's inherently high specificity in terms of potential cross-reactives for the technology. There is, of course, a risk of primer dimers are self-priming specificity issues, which is one of the things you have to guard against when you're designing and developing such a product. It's very rapid amplification. If you set the reaction up correctly and you have a good enzyme and an efficient primer set, the amplification process can be as short as 10 minutes to a positive result from the start of the amplification. And typically the highly processive enzymes which are used in LAMP and certainly in Hibergene's LAMP allow the assay to tolerate significant levels of impurities, which means that quite crude samples can be used rather than having to use classical extraction or other significantly time-consuming or expensive sample cleanup processes. I want to talk just a little bit about how the signal is generated, because at the Hibergene assays we use LAMP with fluorescent signaling, and we have a number of different approaches to this. So what I call our first generation tests, which were the first 4 that we developed here out of our Dublin base, use an intercalating dye. So those are very good. We get a very good strong signal. The fluorescence at the start of the reaction is essentially 0, and then as double-stranded product becomes formed throughout the course of the reaction, the signal is generated at quite high levels. Later on, we move towards multiplex detection, and I can talk a little bit about the test that we have in which 2 different species can be distinguished, differentiated within a single tube. And to do that, we had to look at specific fluorescent probes which allow us to multiplex. And so we use a combination of different probes. Some of them start the reaction quenched and become unquenched as the reaction proceeds. As you can see for the signal below labeled NM, which is actually an example from our pneumococcus, meningococcus combo test. And then others start the reaction at maximal fluorescence and become quenched as it proceeds. As you can see in the signal labeled SP on the bottom left. So these are happening across 2 different fluorescent channels, which we have on board of our instrument. So it's quite a complex process in terms of the signal generation on board the analyzer, which means that we have some quite capable and complex software to interpret those results and ensure that we don't have transient signal events which give rise to false positives. Just a little bit on this slide about the reagents, how we present them. So these are LAMP reagents which contain the fluorescent labels. We present them and we sell them as freeze-dried reaction strips. So these are complete reaction mixes containing the primers, the polymerases, the dyes, many of the buffer components. The freeze-drying process allows us to do a couple of things. It allows us, first of all, to give a reasonable product stability to these products. So typically, Hibergene lamp products are stable for 12 to 18 months post-manufacture, stored at 2 to 8 degrees. It allows us to transport those at ambient temperatures without a cold chain necessity. It also means that when you're reconstituting a freeze-dried reaction mixture with a clinical specimen, you are maximizing the amount of target that goes in to that reaction tube. So it allows us to maximize the sensitivity of the tests. All of our tests are a 2-tube format. So 1 tube contains the target reagents, whether that's for a single target or for multiple targets. And the other tube is a extraction control. And the extraction control validates the negative results. It detects typically an exogenous piece of DNA that we put in the process for sample handling. And the correct detection of that at the end of the reaction validates that the negative samples have been correctly diagnosed. As I mentioned, one of the advantages of the technology as we have presented it is that many of the tests can be performed without significant sample cleanup. So I've just given a couple of examples on this slide of how samples are prepared prior to testing on board the instrument. The Hibergene tests, which range from neonatal detection through STIs, through respiratory detection, by definition work with a range of different clinical specimens, including blood, CSF, stools and urines, bowel for mycoplasma pneumoniae, and then a range of different swabs, which means a range of different transport media. So there are quite a large number of sample types covered by our 13C product range. So the example here on the top right will show the direct testing of bacterial meningitis, specifically Neisseria meningitidis, from a throat swab. And in this case, the procedure is typically take the throat swab, elute the swab in a tube of media, which is presented with the kit, put it on board a heat block for 5 minutes, and then a quick spin as a preparative step before adding that heated spun material directly to the freeze-dried reaction mixture. And typically we use a heat step within all of these processes as the lysis step. So we will typically use heat lysis for bacterial or viral cells, the exception being group B streptococcus, where we do have a enzymatic lysis that occurs at room temperature. And in that case, the heat step actually is to kill the lysis enzyme before it encounters our reagents in the tube. But typically it's a dilute, heat, and add to reaction strips, whether we're talking about a respiratory sample, a stool sample, or a swab sample. Obviously for blood and CSF, for applications such as group B streptococcus, it is preferable and necessary to extract the sample. And we've also validated extraction with simple Keating spin columns for use with our COVID test. But in general, we have very good performance relative to extracted PCR with this relatively crude sample preparation process. All of the tests in the freeze-dried 2-tube format run on our HG Swift analyzer. This is a small, portable piece of equipment. It runs either off main or battery. It will run for 24 hours off a 1.5 hour battery charge. And it's essentially a real-time thermometer with tight control of temperature. So this is a real-time system. So as the reaction is running, as fluorescent signal is being generated in response to the presence of increased level of amplicon, you will see the fluorescent signal change. The software is looking for the maximal rate of change of that fluorescent signal, and when it encounters that, it will report in running the performance to the end user. So a sample will be called positive or within the course of the run. When it becomes positive, we must wait till the end of the run to call the negatives, provided the extraction control has amplified. This can be connected to a small label printer to print the results screen. It can also generate PDFs and CSV files for sample tracing. We spent quite a bit of time working on the software so that we would have something that was intuitive and required minimal interpretation. So on the left-hand side here, you can see an example of the screenshot for one of our tests. It's actually the chlamydia gonorrhea combo test in running. What happens to the end user is, as soon as the sample becomes positive, in this case on 11 minutes for sample 1, that will have been flagged to the laboratory user, so that sample could be reported. And you can see here that we have a range of ways of presenting the result. So sample 1 is positive for chlamydia, sample 2 is positive for both, sample 3 is negative, but sample 4 is flagged as invalid because there's been no amplification of that extraction control. And in that case, our instructions for use contain troubleshooting to point the end user towards where there may have been an issue, either in the sample preparation or potentially upstream of that in some handling of reagents or storage or shipment. So there's not a lot of work to do in terms of interpreting the results here, because our software is looking at those fluorescent signals. It's filtering out any potential false positives due to transient fluorescent signal change, and it's returning positive, negative, or invalid results. We have, as I say, CE marked our range of 13 infectious disease products in alignment with the IVD directive, which means that we've performed head-to-head studies with reference methods for each of these tests. Often that reference method is PCR, but in some instances, for example, GBS, the current gold standard is still enriched culture, at least here in Europe and Ireland. So we performed testing relative to that test method. Nonetheless, occasionally across the 6 years since we've been in business, we have worked with third parties who have published their data. So I just wanted to present a little snapshot of our publications here. As I say, this hasn't been our main focus, but we have worked with with a number of third parties who have published. The meningococcus has been quite well studied. So for 2015, there's a Lancet Infectious Disease paper which reports the testing of 148 pediatric samples using our LAMP primer set with very good PPB and NPV. Then ECCMID the next year in 2016, we have a poster which demonstrates the comparable performance of our meningococcus test against PCR, and also points to that very fast time to positive result, so 12.5 minutes from start of test to positive result return for those meningococcus tests. And just recently this year, we've had a very nice publication using our product. This was an 18-month longitudinal study in the British National Health System across 3 hospitals, testing 260 pediatric patients with query meningitis. And in this case, a throat swab was taken and tested with our simple throat swab sample preparation, which I mentioned in an earlier slide. And that was compared to blood or CSF PCR testing. There was a very good sensitivity and specificity from that testing, which demonstrated the utility, or the possible utility of this test for diagnosis of critical bacterial meningitis without the need for an invasive sample type, specifically blood and very importantly CSF and the associated issues. We've had a number of publications also for our Group B streptococcus test. The Curry et al. Paper from 2018 was a study performed here with the National Maternity Hospital in Dublin, which at that time, and possibly still, was the busiest maternity hospital in Europe. We tested 400 pregnant women there and compared the results to enriched culture and observed very good sensitivity and specificity results in line with the PCR data that's been published for this testing. And critically, the sample setup time and time to result indicated the utility of this test for intrapartum use, so that we could return a result quickly and rapidly to enable a clinician to administer antibiotics to a woman before entering labour. We also have a paper that's been submitted from the German National Reference Centre for Streptococci, where they tested a large selection of their archived clinical isolates, which span 9 different serotypes, and demonstrated 100% detection with our test. And at the moment, there's a publication in preparation in the Mater Hospital here in Dublin, who have performed our third-party independent validation for the COVID test, and they're pulling together a publication at the moment, which they hope to fast-track. I can touch in a little bit more detail on the final 2 slides on the performance of that COVID test. So again, that's just an indication of the level of rigor that we apply internally to validating these tests, and the level of data that's been generated out of the field using these freeze-dried fluorescent lab tests in real clinical situations. I'm just going to present a couple of slides now focusing on our respiratory range, which obviously COVID-19 forms a major part at the moment. But we have tests for Mycoplasma pneumoniae, RSV A and B, flu A and B, which predated the COVID test, and in fact enabled us to get to market very rapidly with the COVID-19 test that we launched here with CE Mark in May. The Mycoplasma pneumoniae test operates from either a throat swab or a bronchoalveolar lavage. It's a reasonably simple sample preparation process in either case. As I mentioned before, typically we take a throat swab or another sample, and there's some minimal cleanup, occasionally a small benchtop centrifugation step just to spin down particulates, then a dilution, and then a heat treatment step. In this case, we have positive results from the Mycoplasma pneumoniae test within 30 minutes. Our sensitivity was 95% relative to PCR, with specificity of 100%. And this test contains an endogenous control. So we have a housekeeping JPDH gene in there, which also controls not just for reagent performance and instrument performance, but also for taking the sample. And as with all of these products, we have done limited detection testing, either against quantified plasmids or, where available, against quantified cell preparations, as you see on the subsequent slides. The respiratory syncytial virus test is similar. Again, starts from a nasopharyngeal swab. In this case, the current version of the test is a dry swab, so the swab is not placed in a transport medium. It comes to the laboratory without transport media and is then eluted in a tube of buffer, which we provide in the kit. This buffer essentially places the eluted virus in the correct salt and pH conditions for the reaction to proceed optimally. We are working for both the RSV and the flu products at the moment on a revised protocol, which will make it completely in alignment with the COVID test. And in fact, we hope to have the flu version of that out by the end of this month. And that will mean the same sample going into a transport media can be tested for COVID and for flu. But for now, the RSV test and the flu test currently in the catalogue, for the next couple of weeks at least, use a dry swab. So here we've tested against multiple strains of the virus, both in terms of LOD and in terms of spiked clinical specimens. And we report the sensitivity limits in terms of TCID 50, because in these cases we're able to obtain quantified material for testing. The flu is very similar. Obviously, in the development of a flu test, the major concern, especially for a LAMP assay, is in identifying a consensus region, which will not be highly variable year on year in terms of the evolution of the flu virus. So this is a difficult design task for this assay, and we've tested it against multiple strains of multiple subtypes of virus. We continue with all our tests, but with a focus on the viral tests, to perform biannual or annual review of the database sequences to ensure that there has been no drift in sequence, which would mean our primer sets are no longer in alignment. Occasionally we come across a low prevalence for some of these products after a number of years, a low prevalence of new strains which may have mutations at certain points. And at that point we will construct worst case, typically plasmid-based models, and test the assay to ensure that we haven't lost sensitivity relative to the current and evolving situation. So to conclude, I really have just 2 slides on our COVID test. As I mentioned, this is something we started in development, I think, at the end of January, start of February, with our R&D team here in Dublin, and harnessing the experience that we had gained from the development of the flu assay and the RSV assay, tried to move as quickly as we could to develop and validate a test. I should say that our RNA-based virus tests are one step, so that we have a single cycle which includes a reverse transcriptase step within the standard LAMP cycle. So it's a one tube, one cycle test. Obviously for COVID-19, we were able to use able to benefit, I would say, from the open exchange of scientific information, and to be able to access sequences early in the process, and to be able to check back during the development process as those numbers of sequences that were databased were very significantly increased. The COVID test is based on an N-gene target. It operates using a nasopharyngeal or throat swab, although we're currently looking at validation of additional sample types, which obviously are becoming quite important and interesting. And importantly, we validated this test in a combination of uses, so using what we call the direct method, which is a non-extraction method, and also in an extracted case. So we do recommend a testing algorithm for this product. What we've identified is that in terms of the non-extracted usage, the optimal way to use this test is when it's close to onset of symptoms or where viral loads are high. So we recommend that if testing patients within 7 days of onset of symptoms, then the direct format is acceptable, and we've demonstrated performance there in the range of 86 to 95%. But it is possible that negative results could be obtained for lower viral loads, which should be retested after extraction. But in any case, if labs want to go directly to the maximal level of sensitivity across the viral load range, we recommend performing extraction. We validated extraction with the Kia AMP RNA mini kit, but we're in the process of extending that validation, and there's no reason why many of the automated systems shouldn't be compatible. In terms of performance of the test, we can see that we've stratified the data from our third-party clinical testing in a couple of ways here. So using that direct testing approach, where the sample is simply taken, diluted, heat treated, added to the reaction mixture, up to CT values of less than or equal to 28, we have 96% agreement with PCR over our population of samples. And that agreement decreases as higher CT values go into the mix. In terms of days from onset, you can see that within 2 to 3 days of onset, we're 96% agreement with PCR, but that agreement level drops off as we go on from day of onset. If we compare that to the performance of the extraction, where we've taken a subset of these specimens and extracted them using that simple spin column methodology, for this population we had 100% agreement with PCR. Obviously, it's a reasonably small population at this point. We have a lot more data now, but this was the data that we used to launch the product. So we put some confidence intervals around that. But essentially, LAMP testing after extraction for COVID gives positive results really, really quickly, around about maximum 20 minutes into the amplification phase. So with a good structured workflow, it's possible to very rapidly get through extraction and testing and return positive results very quickly. And that's it. I'd like to thank you for your attention, and happy to take any questions you may have on the presentation.
Sorry. Thank you very much for the presentation. It's very informative presentation. I want to know, what about the prices of the technology?
Well, I should get our salespeople here because I'm the scientist, but very reasonable is the answer they want me to give. No, the instrumentation is low cost. So in terms of euro here, the instrumentation, that 4-slot instrument is 7,000 euro as a general list price. We're about to bring out in the next couple of weeks a double-size version of that, which will do 8 samples at a time. That's been driven by the COVID demand, and that will retail, I think, at about 10,000. In terms of cost per test, it varies. I mean, we do not sell direct except in the Irish market. So we sell via distributors, and then distributors pass on to their end users. The typical end user price, certainly here in Europe for our tests, is something in the region of 25 euro, 20 to 25 euro per test.
Okay. So we have to talk to BIKO to have low cost here.
Yes, I suggest you talk to them, but don't be too hard on them.
Okay. Well, thank you very much for your informative exposition. I would like to thanks BIKO also for sponsoring with this great exposition. Gracias, BIKO. Gracias al público por estar con nosotros.